further ado, I'm really delighted to welcome Professor Danilo Russo. Danilo is based at the University of Naples, Federico II, and his primary research interests include ecology, animal behaviour, conservation biology, so that's pretty diverse. Much of his work is focused on bats, hence the talk at the webinar today, but he also works on a range of other taxa. And I've known Danilo for several years now, and I often wonder whether he has more hours in the day than all of the rest of us, since his activities, in addition to his research and teaching, are so diverse and numerous. He's a member of the Chiropter Specialist Group for the IUCN. He's vice chair of the Eurobats Advisory Committee. He's a member of several scientific committees. He is editor-in-chief of Mammal Review and associate editor for several other journals. And I'm not quite sure how he does it. But without further introduction, welcome Danilo, and I am going to stop sharing my screen now so that you can share. Okay. Over to you.
Thank you very much, Kirsty. It's a great pleasure to be here today. And well, the answer to your question is I struggle all the time. Okay, I hope you can see my screen now. So thank you very much, and hello everyone. Great pleasure to be here with you, and unfortunately online, but that's what we can do today. It's the only way to keep people in connection. And well, my talk today will be about the use of bats as bioindicators, which is quite a popular topic, but well, there are pros and cons of using these mammals as bioindicators, as we will see during my chat. I would start by mentioning a definition of bioindicator, which according to the Oxford Dictionary of Zoology is any species of narrow amplitude with respect to one or more environmental factors, and that is when present indicative of a particular environmental condition or set of conditions. I have my own definition for bioindicator, which is a little bit broader than this. I would call a bioindicator any biological entity, taxon, or process that responds in some way to environmental characteristics or their alteration. Of course, there are a lot of organisms which may be comprised within this very general definition. Paper of around 20 years ago presented a nice classification of indicators, which was actually classifying them as environmental indicators, ecological indicators, and biodiversity indicators. So environmental indicators are those that can be used to detect a change in environmental state or monitor changes over time or space. An ecological indicator is an indicator which can demonstrate the impact of stressor on a biota or monitor longer-term stressor-induced changes in the biota. And biodiversity indicators are also called surrogate taxa, or those that identify diversity of taxa in a specified area or monitoring changes in biodiversity. We'll come back to these definitions later when we'll talk about bats in particular. So there are issues about selecting bioindicators, and this also concerns bats. First of all, there are potentially thousands of indicators out there, okay? And the selection of indicators is not just an academic exercise, because they do affect policy and management. So they need to be chosen wisely. Using too many indicators may also be confusing, because sometimes indicators may provide contrasting results. And, you know, the selection of indicators may become a sort of new battleground for partisan arguments. So my bioindicator is better than yours. Just to be clear. And I would say that it's also a bit unethical to sell a given taxon as the best bioindicator because we want to protect it, or just because we like it. And you may have heard of, you know, this tendency towards saying that my bioindicator is best just because I want to protect it, and you know, I want to underline an added value of that particular organism to convince policymakers that it would be worth preserving it. but it's unethical because bioindicators are not useful for that. I mean, they are tools that we must use to reach other targets. So we need an objective set of criteria to select bioindicators, and there are some described in the literature. I personally like the procedure described by Rice and Rocher in 2005, which consists of 7 steps. First, to determine user needs. Second, to develop a list of candidate indicators. So I'd like to stress this. The first point is to determine user needs. We are not starting from the taxon, we're starting from the needs, okay? Second, to develop the list of candidate indicators, to determine screening criteria, to score indicators against such criteria, to summarize scoring results, and then decide how many indicators are needed. And finally, to have a selection of them. So we should be honest and say that specialists are generally biased towards their totem species, okay? If I am a bird specialist, I will prefer birds as bioindicators. If I'm a bat specialist, I will think about bats as fantastic bioindicators in all situations, which is not always the case, to be honest. A little bit of history, it's recent history, really. I think a turning point in the scientific discussion about the use of bats as bioindicators was represented by a paper by Garth Jones and colleagues published in Endangered Species Research, this Carpe Noctem, the importance of bats as bioindicators, which for the first time gather all the evidence to support the idea that bats could be useful indicators in many cases. Then we had a very nice international symposium on importance of bats as bioindicators in 2012, if I'm correct, in Catalonia. And later on, Garth Jones and myself co-edited a special issue of Mammalian Biology on the use of bats as bioindicators. So let's see whether bats are useful bioindicators. Bats are already used as bioindicators in certain countries. For example, in the UK, a set of species, of bat species, is used as bioindicators, and their trends are followed over time. And the UK may count on a fantastic network of operators in the field, volunteers and so on, so they can monitor bats very closely, and they have data which can be used to draw trends over time of bat populations. Here to the right, you see what's going on to the bat populations in the country, 1999 to 2019. There's a nice increasing trend. This is fantastic. I mean, if you want to see what's going on to community, biological communities, and you select focal organisms to do that. The problem with the very concept of bioindicator here is that it is difficult to relate this trend to specific factors, environmental factors, which are affecting these trends. And this is a sort of general problem, as we will see later. Paper published several years ago, again in the British Isles, England and Wales, if I'm correct, looked at the Benton's bat distribution and abundance, and developed a predictive model to use this species for bioindication. The Benton's bats are especially useful from this perspective because one of the desirable properties of bioindicators is that they should be strictly associated with a certain habitat type, which is not the case with many bat species. But in this case, the Bentons are strict waterway and lake specialists. So if you want to use the Bentons as a bioindicator for water quality, river quality in general, I mean that their specific ecology is a pro, certainly a pro for to reach this goal. The terms used for the model were biological water quality, waterway width, and mean annual discharge and presence of trees. So both biological and structural properties of rivers. And it was seen that the activity of this species was predicted to be higher on larger waterways with abundant woodland nearby, but there was a lot of site-specific variation, which is obviously a constraint towards the use of this model to bioindicate the quality of rivers. The activity of this bat was predicted to be related to aquatic macroinvertebrate diversity, so and also good chemical water quality. So the activity could be predicted from habitat and water quality data, but again, the precision was low. Limitations. In 2010, the meeting of parties of Euro-BATS agreement is the Convention on the Protection of European bat populations. It's a UNEP agreement. With a resolution 613 urged parties and non-party range states to develop national, regional, and pan-European bat biodiversity indicators, facilitate the incorporation of bat data within multi-taxa indicators, and support the objective of gathering the data for these indicators, as well as to forge cooperation platforms to facilitate data exchange. So the use of bats as indicators became, in that situation, a well-established policy objective, I would say. And Eurobats was behind the development of prototype indicator for European bat populations, which was funded by the European Environmental Agency. And in this case, the index was based on 9 countries and 16 species. The countries selected were those for which hibernacular monitoring programs were available and sufficiently reliable. And here to the right, you can see how this index worked. So they started from identifying national trends and indices for the focal bat species. Then these were lumped together into regional trends and indices for bat species. And then at a higher level, these were pulled together into European trends and indices of bat species, which were summed up finally in a single European indicator of bats. And the overall trend was increasing. I mean, many species were showed to be on the increase, probably because of better protection, possibly. But again, this indicator is useful if you want to see in a synthetic way what's going on to European bat species. But it's not that useful if you want to see what's going on to the environment and what are the environmental factors that determine this increase. Anyway, the index should be developed further, including more countries and maybe more species. And again, the absence of direct relationships with environmental factors is an issue from the pure bioindicator viewpoint, if you see what I mean. So just for the sake of this talk, I did a very short, very quick Scopus search. And I used bats and bioindicators as keywords, and I retrieved 103 documents, 42 of which really address the topic in some way. Many others were not strictly relevant. Sometimes, papers just mention the fact that bats may be useful bioindicators, but they do not use bats as bioindicators really. And other papers were on monitoring techniques. These papers mostly looked at the use of bats as potential bioindicators. So they concentrated on the monitoring side of the story without exploring the effectiveness of bats as bioindicators. And I mean, my overall impression, just going through these papers, was that we are still very far from establishing strong and sort of continuous research lines on the topic. So let's go back to the Garth Jones and colleagues paper on the importance of bats as bioindicators. These are the main points that these colleagues highlighted as the characteristics of bats which would make these organisms good bioindicators. First of all, taxonomy of bats is relatively stable. Bats can be sampled at several levels, from populations to feeding rates. Many species do occupy a wide geographic range, and in many cases bats respond greatly to habitat degradation, and these responses are correlated with responses of other taxa. For example, many bat species are insectivorous, so whatever happens to their insect food is reflected into bat populations. But bats are also characterized by a rich trophic diversity. There are bats eating several kinds of food items, not only insects, but also fruit, pollen, vertebrates, and so on. So they can be representative of changes in the environment at different levels. And they have a slow reproductive rate, which means that it takes time for bats to recover from any impact of environmental stressors. They perform key ecosystem services such as pollination, fruit dispersal, arthropod control, and they're also reservoirs of wide range of emerging infectious diseases whose epidemiology may reflect environmental stress. So these are this is the profile of bats as potential good bioindicators. Let's go and look into some of these points. First point is whether bats are that stable from the taxonomical viewpoint. I mean, they're way more stable than other organisms. Think about what happens to mollusks, for example. But still, we know that in last few decades, thanks to molecular studies, many new cryptic species of bats were described. The slides just concern some of the bats in the European bat fauna, which were sort of strongly changed by new arrivals, as it were. Okay, the Plecotus group, for example, or the Myotis genus. We had a lot of new species which were discovered thanks to molecular approaches. And of course, these may, at least locally, undermine taxonomic stability. Just to make an example, in the UK bat bioindicator, whisker bats and bronze bats, if I'm correct, they are pulled together just because they are difficult to distinguish. They are cryptic species, and in many cases, only molecular analysis can tell species apart. So this is a limitation which cannot be ignored. Sampling bats is not easy. I mean, I think all of us here are bat specialists, and we know that catching bats is complicated. They are elusive, they are nocturnal, and they're also sensitive to disturbance. So it's not really easy to carry out direct observations of bats. But we have a fantastic approach for bats, which is the acoustic techniques. If we use acoustic methods, we can rely on a non-invasive methodology to survey a lot of species. Of course, we'll miss some species, because some species broadcast faint calls or very directional calls, so they are overlooked in acoustic surveys, but still, we get a lot of species. Anyway, identification problems are still there, and we know very well that it's practically impossible, especially in rich communities, to provide 100% confident identification of all species present. And there are costs too. If you want to rely on good tools for acoustic techniques, the costs are quite high. We must say that some of these issues are being, if not solved, at least mitigated, I would say, by advancing technology, for example, ID problems or costs, okay? But still, we cannot deny that identifying bats in field based on recordings is still an issue. Of course, we have automatic recording, which is fantastic. We can store a large number of recordings, and this will help a lot in the use of bats as bioindicators as well. But again, the problem of acoustic techniques is partly still there, although mitigated by the advancing technology. Then I'd like to mention another problem which bats pose in terms of presence-absence. Because bats may get overlooked, I mean, we can miss species. Pseudo-absence is always behind a corner. The species I shown in the slide here is the Baxland bats, European species, which is way less rare than it was thought. I mean, it's present in many European forests, but it's a faint color, it's Myotis species, so its calls are difficult to identify, and it's very difficult to catch. So in many places, you think that there are no Baxland bats around, but they're there. So pseudo-absence is a serious issue for this species in many countries. And presence, especially for bats, does not equal preference. Ideally, a bioindicator should tell us something just because it's there or it's not there, okay? But in this specific situation, in situation of bats, you can find bats in several habitats, and still this will not tell us whether the habitat is favorable to them or not. This is a problem even with specialist bats. Bats are multiple habitat specialists. They can use several kinds of habitats. And from this perspective, their powers by indicators may not be fantastic. I have an example, it's very close to my heart. It's the barbacil bat. We've been studying these bats for over 20 years in Italy. And when we started to work on barbacils, we found ourselves in the ideal situation. We found these bats to thrive in unmanaged forest with a lot of dead trees. So fantastic situations. And you could, come up with the idea that this bat is really a marker of that kind of forest. Then we found this bat in very different situations, in log forests, for example, or even in areas where you had no forest at all, in clay badlands in central Italy. Where these bats actually may reproduce, roosting in crevices, in rocks. And I mean, they are just very happy because you have plenty of moths in those areas, which are the staple food for this species. So at least if you want to use this species in terms of presence as a marker for this well-established kind of forest, or stage of forest, well, this species will just not work, I would say. One of the problems bats pose as bioindicators is that they are indeed very sensitive to environmental alterations, but being multiple habitat users, their responses to stressors, to environmental stressors, to habitat change or degradation, may be related to different factors. I mean, bats are sensitive to foraging habitat quality, to food availability, to water availability, but also to roosts, okay? So you may have ideal foraging habitats around, but if you lose roosts, bats will disappear. And commuting routes are also very important. I mean, bats may cover very long distances from their roosts to foraging sites. And if these routes are fragmented, then you will experience declines in the presence of bats or in their abundance, which are not strictly related to the availability of food or foraging sites, roosts, but just because moving across the landscape is way more difficult. And generally speaking, bats are sensitive to a range of environmental factors which act at several spatial and temporal scales. Let's take the situation of the effects of urbanization on bats, for example. We know that there are endangered species, threatened species of bats, which are jeopardized by the expansion of urban areas, okay? Because they lose ground, they lose natural habitat, and they are pushed away from many areas. If you look at species diversity versus the amount of urban areas, for example, in a certain buffer around sampling sites, in many regions of the world, I think, you'll come up with what we found for Italy, which is shown in this graph, where you see that there's a decrease in bat community diversity when you increase the amount of urban area available. So, I mean, simply put it by saying that urbanization decreases the diversity of bat communities. So the thing is, again, urbanization is made of a set of complex stressors, I would say. And if you see that bat communities are richer where urban areas are more developed, you don't really know what factors are behind this loss of diversity. Artificial illumination is one of the candidate factors. There's a lot of fantastic work done on bats showing that artificial illumination is a big threat to bat conservation because it threatens bats at several levels. Okay, we did some work on bats and light pollution as well. We found, for example, that light pollution over rivers is a problem for foraging bats, and bat pollution is a big problem for bats drinking, both in forests and in deserts, so in very different landscapes and regions of the world. So disentangling the factors that are behind the suitability of habitats and landscapes for bats is always a problem, and urban areas make no exception. Let's look at 3 common species in Europe and Italy in particular that dwell in urban areas. Cool pipistrelle to the left, Savis pipistrelle center, common pipistrelle right. So these are urban exploiters. These bats are found almost everywhere in cities, for example, in my country. But still, when we looked at the bat activity, these 3 species in urban lakes, so in blue spaces in the city of Rome, we found that there were very different responses depending on the amount of woodland which surrounded these sites. Okay. So again, sort of subtle aspect of landscape, how much woodland you have in a 1 kilometer radius may affect heavily the presence and abundance of these species, even if these species are tolerant to urbanization. So you would expect them not to be as sensitive to the availability of green spaces within urban settlements. And speaking of bats in urban environments, bat populations, or at least colonies, may respond negatively to factors that are easily overlooked. Think about predation by domestic cats, for example. Okay, they can locally even push an entire colony into extinction. And in that case, if you don't consider cats as the stressor, and that may be easily overlooked, then the resulting picture might be misleading. So bats are important mammals in urban areas. This is a paper we published with several colleagues 3 years ago in Ecology Letters, looking at the richness of mammals in urban habitats. And we found that bats are actually the first group. Okay, about 78 species, if I'm correct, are to different extent present in cities. It's not surprising because bats are also very rich mammal group. But still, I mean, the fact that we have so many bats in cities and so many bats that cannot use cities, that are filtered out by the urban habitats, will tell us that we might try to use bats as bioindicators in our urban environments. So let's try to hypothesize different fitness responses in bat species along urbanization gradients. Okay, so the A condition is what would not be particularly useful for a bat to be a good bioindicator. In this case, you have a bat which shows no fitness variation from rural through suburban to urban habitats. So no response to urbanization at all. Then you have responses which may be hypothetically very clear. The response is sort of urban exploiter, which increases its fitness from rural to urban. And on the opposite side, the B responses of a bat which is very sensitive to urbanization would be something like you have very high fitness in rural, and then this will decrease through suburban down to urban. And finally, we might expect a peak in fitness in suburban situations. Bats that are adapted, for example, to rural environments might show this kind of patterns. Okay, so if we identify this kind of, say, fitness responses in different bat species, then you might select a set of bat species as bioindicators of urbanization. Theoretically speaking, of course. What about farmland? There are studies comparing the responses of bats in organic versus conventional farm, but results are contrasting. There are 4 studies showing more bat species or higher foraging activity at organic sites versus conventional. 1 study showed that few bat species were more abundant at organic sites versus conventional sites, and 3 studies found no difference. Again, here, one of the problems is that the effects are difficult to disentangle. When we say organic, we actually say nothing. Is it pesticides? Is it hedgerows? Is it mechanization used in farm management, and so on? There are so many factors which may potentially influence bat activity, and these effects are really difficult to disentangle. And there's also a problem of landscape versus habitat management. Maybe you will look at a certain plot which is managed in a certain way, and then what you see in the plot is actually the effect of what's going on in the surrounding landscape. So controlling for the effects of landscape is also, from an experimental viewpoint, quite complicated. I found one paper published in 2018 which tried to solve this issue. In this study, which was set in Canada, in soybean fields in Canada, the authors, what the authors did was to select plots which had a equal landscape situation surrounding. So they did control for landscape structure. And in their pair design, whatever effect was highlighted, it was a genuine effect of habitat and not sort of byproduct of the surrounding landscape. In this situation, it was clear that both species richness of bats and bat activity for several species were higher in organic versus conventional soybean fields. And same kind of response was found in insect food, in insect prey. So as you can see to the right, insects were way more abundant in organic than in conventional soybean fields. Okay, so this is the relationship the author found between insect abundance and dry weight and bat activity. You can see that there's nice positive correlation, although the R-squared factors are not very high, which means that this relationship actually captured only a small part of the total variance. So the question is, what other factors may be acting to influence this difference between bats in conventional versus organic fields? One aspect might be the direct effect of pesticides in bats. Bats are sensitive to pesticides, are killed by pesticides, we know that. So apart from the indirect effects of pesticides on bat food, bats may be intoxicated and killed by pesticides. And although there are studies in this, it's a bit surprising to see how this aspect was sort of neglected, also in terms of policies. I was fortunate to be part of a EFSA panel. EFSA is the European Food Security Authority, which was devoted to work on the coverage of bats by the current pesticide risk assessment for birds and mammals in the EU. And what we concluded actually was that bats are not adequately covered by the current risk assessment approach. So there's a need to develop a bat-specific risk assessment scheme. It's the first step towards more work, which should lead us to consider comprehensively, at least in the EU, the bats as a biodiversity component that is heavily affected by pesticides. Let's talk very quickly about forestry. I think that forestry is probably one of the most promising fields where bats might be useful bioindicators. There are several papers showing that bats respond differently to different silvicultural treatments. I took part into a study a few years ago. It was part of a EU LIFE project in which we measured responses by bats to different silvicultural treatments. I'm not going to go into the details of the study, but as you can see from the graphs to the right, we looked at what happened in control versus innovative and traditional logging procedures. They differed in terms, for example, of amount of dead wood which was left in place, distance between trees, and so on. So if you look at what bats will do, under different treatments, you will find that there are quite clear responses in terms of bat activity to these different procedures. And what I found especially interesting in this case is that even just looking at the overall activity of bats was very informative, which is fantastic because you don't really need to look at specific species. So you overcome, all the identification problems. You can just leave bat detector in the field, measure total bat activity, and you will have a response which means something in terms of the effect of the treatment. River pollution, another, I think, promising field where we might propose bats as useful bioindicators. There is evidence in the literature that bats do respond to river quality in terms of richness and activity. I already showed you some studies which suggested this might be the case, although with low precision, with low resolution. But there are other studies which are sort of more positive on the topic. This is a paper published a few years ago in the US, which looked at the effects of water quality and urbanization on temperate insectivorous bats. And the focus here was the landscape scale. And what the authors concluded is that actually bats responded clearly to different water quality and also to urbanization at landscape scale. So again, bats as potentially useful indicators in this case. In the Iberian Peninsula, there's an index based on Meiotis del Bentoni presence and activity. It's called the Quiroreus index, which is pretty like the index I mentioned earlier, used for the UK. And in this particular study, the authors set out to explore correlations between this the Benton's bat index and other two common indices which are used to characterize river quality in the Iberian Peninsula in Spain. One is related to macrobenthos and the other to vegetation. Okay, so one is more, say, related to the biological side, and the other is more related to the structural side of the river in terms of vegetation. The key point here is that there was little agreement among these three indices, which question use of each single index for bioindication. And the authors concluded that the three indices should be used together to characterize water quality comprehensively. A study we did in Italy in 2018 was somewhat more optimistic because we did something very similar. We looked at two indices. One is the IFF, is the river function index, and it's sort of a complex index which takes into account several components from biotic and abiotic components taken together, okay? And it gives you a sort of overall picture of water or river quality. And then we looked at water quality, examining the macrobenthic communities using the STAR ICM index, which is the official index used in the country. In this case, we found that both richness and especially activity of different species responded clearly to the two indices. So we did find correlations, regressions in this case, which would allow us to use bats to characterize water quality and river quality in several situations. Of course, our study was just set in a limited geographic regions in central and southern Italy, so it was difficult to generalize to the whole country, and we will need more replications to come up with generalizations. But I think that at least that was a first step towards convincing use of bats as bioindicators in river habitats. So what about bioaccumulation? Here, I think the game is easier because bats do bioaccumulate chemicals such as heavy metals and pesticides. We can bioindicate presence of these pollutants just by looking at bat tissues. Even non-invasively, fur, for example, is very useful for that. And if we have to look at organs, we don't have to kill bats because we can, for example, cooperate with rehab centers where unfortunately many bats won't make it, and but there is a lot of material available for this kind of analysis, which might be used. also, you know, in a systematic way across countries or large regions. There's a nice study set in Germany, which actually showed the effects of remediation treatment in a small lake, which was affected by presence of heavy metals. So what was done there was actually to dredge the sediment and clean up the place, basically. So the authors analyzed the content of heavy metals in the Bentonsbad soil before and after the treatment, and they found actually that the concentrations of these heavy metals decreased after the treatment was performed. I think it's a very nice story which tells us that bats can be effectively used as bioindicators of heavy metal presence in the environment, because they react promptly and are very representative of whatever happens in the environment. Climate change. Bats have been long thought of good organisms to indicate climate change because they are sensitive to temperature and water availability. So two key points that are, you know, changing or likely to change further in the future in relation to climate change. And this study published by Newson and colleagues showing proposing Adon album and maximum free-tailed bats as potential organisms to be surveyed for their potential responses to climate change. We know that when it comes to climate change, most of the scientific discussion on bats is based on what's going to happen. So there are predictions based on modeling, but there are a few bats which are actually already responding clearly to climate change. And cool spittle are one such example. This species in Europe increased its range 400% times in the last 40 years. Okay, so we had a 400% range expansion in the last 40 years. And according to our work, the most plausible explanation for this expansion was actually the changes that European climate experienced in the same period of time. We did some modeling work using presence data, past presence data and current presence data, and we found that climate change was the best candidate to explain this expansion. So can we use bats as indicators of climate change and its effect on the biota? The answer is possibly yes. I had the fortune to chair an action, a EU coast action, which is called Climate Change and Bats from Science to Conservation. We have three main objectives. One is to assess and predict the impact of climate change on bat distribution. The other is to establish networks to monitor changes in bat distribution and populations. And finally, to assess pest predation by bats and predict what's going to happen to this ecosystem service because of climate change. So say that one of the objectives we are pursuing is actually to use bats as indicators of climate change. Finally, can we use bats as surrogate taxa? Surrogate taxa are, as I anticipated, groups of organisms whose species richness may be representative of the broader species richness in biological community. And they are fantastic, ideally, because you just have to survey these taxa to get an overall picture of biodiversity of a certain habitat or area. So think about, for example, the need of identifying quickly protected areas in rapidly developing regions of the world, instead of devoting a lot of efforts into monitoring many biodiversity components. You just look at a few of them and you have a picture of the overall richness. So the example to the right is an old study done in Austria in farmland, showing how certain organism groups can be used to portray the overall species richness in this kind of habitats. You can also pull different taxa together. It's so-called shopping basket approach to strengthen the effectiveness of your bioindicator. In this case, for example, you can combine vascular plants and bioindicators to come up with a more powerful representation of the overall community diversity, richness. There's a surprisingly small number of studies of bats exploring the potential bats have as surrogate taxa. I found two, maybe I missed something, but I found two studies. One done in Denmark, which is quite promising because actually in this case, both bats and large moths appeared as the most robust taxa in selecting grid cells which feature the highest richness of other taxa, which means that if you look at bat species present there or moths, you can actually have an idea also of how rich the butterfly communities, the amphibian reptiles communities, the click beetle communities, and so on. And another study which was done in Southeast Asia was way less positive on the potential use of bats as surrogate taxa, as a surrogate taxon, because the results were quite contrasting and ambiguous, at least at the spatial scale considered for that study, which was 100 per 35 kilometers grids. Apart from the environmental set where we mean to use bats as bioindicators, a big problem might lie in the say speed, in the promptness, which is shown by bat responses. From our bat specialist experience, if something happens in the environment, normally bat populations will quite rapidly decline. Okay, this is reflected in many studies published. But I also found a study done in Japan, which would show otherwise. In this case, the authors found sort of time-lagged responses to environmental alterations. They found that certain bat species which were lumped together because they work with, I think, heterodyne bat detectors, so it was quite a coarse grain analysis actually, but they found that this group actually reflected the situation of the former forest which used to be present in the study area in 1950, even if much of that forest was disappeared and replaced with farmland nowadays. So there was a sort of depth of time-lag responses to environmental alteration, which would question the use of bats, at least in that particular situation, as bioindicators. But this study was honestly highly correlative, and in my opinion, did not look at other factors which would have been important. So we need more work on this topic. But I think one of the important points of this study is that the issue of how prompt bat response to environmental alteration is an important aspect which should not be overlooked if we wish to use them as bioindicators. And it's something we should test more. So to conclude, what is the way ahead? My first point is that monitoring bats is mandatory in many countries. If you just think about the situation in the EU, for example, to respond to policy requests in the EU, we have to monitor all bat populations virtually. So since we have to monitor bats anyway, it would be wise to develop a strategy which can achieve the goal of using bats as bioindicators, at least in certain habitats where this may be promising, rivers, farmland, for example, and comply with these low requirements. It's a very effective way to reach two goals with one effort. We still need to develop robust and easy to use indicators. So we should concentrate on this aspect of bat studies, I think, to improve bat indicators in terms of robustness and easiness of use. And of course, standardizing sampling methods, at least within a certain indicator of habitat, is of key importance to move steps forward. we might think about using ad hoc indicators. I'm not very optimistic about finding, you know, sort of global solution in a certain, even in certain habitats, because bat communities change worldwide and local situations are different and so on. But we might try to find at least local situations or habitat-related situations which can be explored successfully using certain bat species, certain sets of species, or maybe the overall bat activity if we can. We have a big problem because I think we don't have enough replication of studies. I mean, if you look at literature, you see that there are a couple of studies, 3 studies, 4 studies on certain subject, certain habitat, but we don't have a replication of studies which can really represent the convincing arguments for using bats as bioindicators. And the constraints, I'm sorry, but they're also posed by the academic pressure for novelty. As academics, sometimes we look for, you know, novel research lines, and if we see that something has been done elsewhere, we are sort of discouraged from replicating the study in a different geographic set, which would be absolutely important in this particular situation because it would be sort of confirmation, for example, or further testing of the use of bats as bioindicators in certain context. We also have to better understand whether we can use bats on large scale versus local scale as bioindicators, as I anticipated. So keeping an eye on the value of scale, on the influence of scale in these situations is always very important. And I think it would be absolutely vital to join forces. For example, pan-European or global project. I know I'm dreaming, but a global project on the use of bats as bioindicators in farmland, for example, would be absolutely fantastic to have a better idea of whether, to what extent, we can use bats as bioindicators, and probably, you know, bats might prove that way better than other organisms already used as bioindicators, and gain sort of global recognition of their values as bioindicators as well. Okay, so I think that I'm concluding now. I don't know why, but the presentation got stuck. Anyway, I just had a final slide with nice picture to thank you for your attention. And I'm here for your questions. Thanks very much.
Thanks very much, Danilo. That was absolutely fascinating. We do have a couple of questions coming up in the chat. I'm going to take host prerogative here and ask my own question first. I looked at this topic specifically in agricultural areas and specifically around the issue of the surrogate species. So what are bats actually indicating? Are they can we look at bats and that tells us something about other taxa, as you mentioned earlier in your slide? And it seemed to me at the time, this was several years ago now, that it wasn't that they weren't necessarily useful, but just some of the key studies haven't been done. And they haven't necessarily been done in the way that allows us to answer the question about whether or not that's... So do you have a kind of idealized, in your perfect world, what sort of study would you do that that would enable you to address the sorts of questions that you've been asking here?
Well, you know, research is a tricky field. You know, it's very well. I think that most of the evidence that bats can use as bioindicators comes from conservation biology studies. And this is also due to the fact that we find more funds for conservation than we do for other aspects, for example. So one of the things we should really try to do is sort of you know, see the problem from the opposite perspective. Say you don't start from the necessity of preserving the species and identifying the factors that will influence the species, but on the other way around. I mean, you should really try to study what factors will influence or not the species behavior, and whether the species behavior is sufficiently representative of a response to environmental stressors. But this would need a lot of standardization, and I think more funding, which I'm not sure is currently available. Because
okay. Maybe I was thinking fantasy world more than reality. Okay.
Yeah, I mean, in perfect world, I think, as I concluded, I think we should really join forces, come up with a sort of horizon project or something like that, in which we can really set the procedures and identify the actors of the study in different regions and come up with something which is way more standardized.
That sounds great. Okay, so starting from the top, Rachel's asked a question about, I mean, I noticed that you did have fruit bats in one of your papers when you were talking about contaminants. And Rachel's asking, in regions where they have fruit bats, have they also been used in bioindicator studies, or has the focus been more on insectivorous bats?
No, I think there's a bias. I may have missed something, but the bias is towards insectivorous bat species.
Yeah.
But I think that there's a lot of potential in fruit bats as well. So again,
okay, and then there was a follow-up question to that, saying that since some of the larger fruit bats are relatively more abundant in cities, what are the possibilities to use them as bioindicators, and especially to understand the effects of urbanization?
Absolutely, yes. Absolutely, yes. There's a recent paper on bioaccumulation of heavy metals in flying foxes, for example, which
okay. Thank you. The next one said, not exactly a question from Asol on the topic of bioindicators, but since you've touched on, oh, wait a minute, it's disappeared. Since you've touched on adaptability, what's your opinion on the practice of forced relocation of bat colonies in urban environments?
Well, I'm not a fan of relocation in general. I think it's very difficult to achieve. So it's something that, if desperate, we can try, but I don't think it will go any further.
Okay. Question from Elisa. Are prompt responses, you talked about time lags earlier, are prompt responses perhaps reflecting, oh God, it keeps moving again. Right, sorry, I'm going to scroll. Are prompt responses perhaps reflecting short-term behavioural changes? So bats shifting roosting or foraging areas, and time-lagged responses might reflect longer-term population changes.
Yeah, I think that's one of the key points. We should try to identify different time scales for responses, because they may tell us different things, basically. Another good point for further exploration of the topic.
Okay, then we've got lots of thank yous. Great talk.
Thanks.
Let me scroll down. Food for thought. Thanks a lot. Awesome. I tried to find a question now. Ines has a question. I've been denied funding for bat monitoring projects, as granting agency are typically pressurising the novelty, which you mentioned towards the end. Do you have any suggestions of how we sell these types of projects better?
Sorry, suggestions for what? I didn't hear you.
How we sell what might be essentially monitoring projects, how we try and sell the academic novelty of them better. I mean, I guess that's the holy grail, really, isn't it?
Yeah, but to be honest, it would be very difficult. I would love to be able to answer this question. It's really difficult.
I mean, I think it is. I've seen really nice examples where, you know, people are essentially wanting to continue long-term monitoring, and it might feel like reinventing the wheel every now and then, but just finding that actually we can ask a really interesting science question within that which hasn't already been addressed. I think that's trying to do it can be hard, but that's the way of selling it to a funding agency, I think.
Yeah, possibly.
Okay, I think we are done for questions I can see, unless anyone wants to unmute and ask a question now. You are, maybe if you raise your hand first. No, I think they've all been stunned into silence. Lots more thank yous coming up in the chat.
Thanks very much. Thanks very much for being here.
Okay.
Thanks for the invitation.
And another thank you. Thanks so much, Danilo. That was really appreciated. And thanks very much to everyone for turning up today. And remember to check the YouTube channel if there are other talks that you might have missed. Okay.
Thank you very much again.
Bye.
Thanks. Bye, everyone.
Bye-bye.